Let's begin. Let's begin the tissue lab. You should have your worksheet in front of you. Let's first talk about the four types of tissue. There are four types of tissues found in man, and they are epithelial, connective, muscle, and nervous. We'll be looking at all four of these major types and then different examples of each type in this lab. Number 2 on your worksheet said which tissue type has this function. Which tissue type conveys electrochemical impulses? That would be the nervous tissue. Which tissue covers, lines, and secretes? That would be epithelia. Which tissue binds is found in between tissues and under epithelia. So let me see, it must be between two tissues so it connects them, so it would be connective. And then the last one, movement and support, of course, is muscle. So now let's go into more detail with each type of tissue, starting with epithelia. When we name epithelia tissue that covers, lines, and makes up glands, we name it either by how many layers of the cells there are or by their shape. So let's start with layers. If we have one layer of cells, we call that tissue simple. If we have more than one layer, we call that tissue stratified. You can see this one has 1, 2, 3, 4, 5 layers, so it is stratified. Another term we sometimes use dealing with the layers is pseudostratified, which means falsely layered. Looking at the tissue, you see nuclei at different levels, so you think there's more than one layer of cells. But then if you look closely, all of the cells go to the basement membrane, and so it looks like it's more than one layer, but they actually all come down to that basal membrane, so it's pseudostratified. Another way we name epithelia is by the shape. The first one at the top is short and squatty, is a squamous epithelial cell. The second one is about as tall as it is wide, so we call it cuboidal. And the third one is taller than it is wide, like the columns on the front of a house, so it's columnar. There is another shape name we use sometimes, and it's transitional. I drew a bunch of simple squamous cuboidal cells surrounding this space, and that's supposed to be my organ, maybe like the urinary bladder. When the urinary bladder is empty, those cells look cuboidal, as tall as they are wide. But as my organ fills up with urine, my epithelial cells are stretched, and now they look more squamous. So we call cells that change their shape as the organ that they make up changes transitional, because they change. So where would you find epithelia if you were looking under the microscope? Epithelia covers, so you would find it around the outside of an organ, or it lines cavities, so you would find it inside of the cavities. You also find it making up glands. So let's review the words used to describe epithelia. Simple, which one does that match? B, single layer of cells. Stratified. C, 2 or more layers. Pseudostratified. E, one layer that appears multiple. Transitional. G, appearance is variable as the tissue stretches. Squamous, and I have too many O's in squamous. Flat, scaled-like, irregular cells. They're short and squatty. Cuboidal, tall as they are wide, so that would be A, block-like. And columnar are tall cylindrical cells, columnar. So these are the words that we use to describe epithelia. So let's put some of these words together and look at the types of epithelia. If you notice the artist view up in the upper left-hand corner, there's one layer of flat scale-like cells. So this is simple squamous. If you look under a microscope slide, you'll see that they're very thin and they just outline the spaces. So on simple squamous epithelium on your little chart, you can just say a line around a space. That's a pretty good description. Or if you want some other description, you can put it there as well, because these descriptions are going to help you identify it later. Let's look at another epithelia. We're actually going to the third one in your chart. This is one layer of block-like cells, so it's simple cuboidal. Where are we finding them on the slide? Well, each one of those little round circles is one layer of cuboidal cells, and you see the space in the middle. It kind of reminds me of wagon wheels, so you can write what description you want down that'll help you remember this is what simple cuboidal looks like later. Let's go back to the second one, simple columnar. A lot of people say these look like teeth and the mouth in between. These are actually two different layers of simple columnar cells. One layer of taller than wide cells, simple columnar. And you can write your description, maybe your teeth inside your mouth. This is not simple because we have multiple layers. This is stratified. We name stratified tissue by the type of cell that's at the very apical end, or the end that's to the air space. And when we look at the air space at the top of this microscope picture, we can't hardly tell anything about the appearance. They're so flat and scaled-like and squished, so they must be squamous up there. So we would say this tissue is stratified squamous. Oh, and as far as appearance, I'm not sure what you want to say about it. To me, it looks like skin because I know what skin looks like, but you just put your own description down there. So let's move to the next one. We could say ciliated stratified columnar, or actually the one that we're seeing in this picture is pseudostratified. I do have stratified ciliated columnar, but when you look at the artist view, you can see that all of the cells are sitting on the basement membrane. They don't all extend to the apical surface, but they're all sitting on the basement membrane, and so the nuclei look like they're at different levels. At the apical end, we have hair-like extensions called cilia. So this would be stratified cilia, or more precisely pseudostratified cilia. I don't know, to me it looks like the ends of a brush. Let's move on to connective tissues. Connective tissues have three things in common. They have cells that are spread out, there's a matrix substance in between the cells, and in the matrix you have fibers. Connective tissues have the most variety, they're the most distributed, they look more different from each other than all the other tissue types. Let's look at this first connective tissue. When I look at it, I see a lot of airy spaces in there, so it's areolar. It's called loose because the fibers aren't stacked one on top of each other, but they're all spread out. You find loose areolar connective underneath all epithelia as part of the basement membrane. Here's another loose connective tissue. You see those big white blobs that kind of look like marshmallows? Those are fat globules. This is adipose. It's loose because the fibers are all spread out away from the fat. You can't really even see them very well. So this is adipose. It is a loose connective tissue. This is blood. You can see the larger white blood cells, the smaller red blood cells, and the teeny tiny platelet. Those are the three kinds of blood cells. This is a loose connective tissue. Actually, the fibers are dissolved right now and won't become visible until blood clots. So this is blood. I don't know if you want to put polka dots or how you want to describe it, but blood. This one's easy to describe. It looks like the inside of a tree. It looks like you have the rings of the tree. And that's the way bone or osseous tissue is made. It's made in osteon units, concentric circles to give bone strength. So this is bone tissue. Here's another connective tissue. This is cartilage. The little cartilage cells are called chondrocytes, and they're in spaces called lacunae, and they have a plastic matrix in between. To me, it kind of looks like a beehive, or a place where bugs have burrowed in and they've laid eggs in these little separate sacs. I'm a biologist, so that's probably why my mind goes there, but you can describe it or how you want to remember it again. Hyaline cartilage. This is not a loose connective tissue. Do you see all the fibers are very packed and they're running in a regular manner? So we call this dense, regular, white connective tissue. And you find this in ligaments and tendons. It looks like waves. Now we're on to muscles. There are three kinds of muscles. This is cardiac muscle. It is an involuntary muscle. It is striated. You see the little stripes. It has a central nucleus. They are short fibers that branch, and they are responsible for moving blood through the heart. Kind of looks like bamboo. Here's another type of muscle. This is skeletal muscle. It also has fibers, but they don't branch. They're very long. They are striped or striated. It's the only voluntary muscle. It doesn't have one nucleus, but is multinucleated, and the nuclei are pushed to the outside. Someone told me it looked like logs on a log cabin, but you can describe it the way you want. And it does move bones and gives you facial expressions. The last kind of muscle to me doesn't even look like muscle. It looks more like epithelia. The cell, if we could isolate one cell, it would be fat in the middle and have pointed tapered ends. We say it's spindle shape. It is involuntary. It has one central nucleus. It's found in hollow organs to move substances through the hollow organ. And it is non-striated, hence its name smooth. I don't know, this kind of looks like wood to me, or material, but you describe it the way you want to. And we're off to the last tissue type, and that is nervous. Those brown cells, they kind of look like balloons or kites on the end of a string. Those are the neurons. Those extensions are either dendrites or axons. All the yellow material behind it are neuroglial cells. The artist drawing is of what a neuron looks like. Those extensions are dendrites that bring an impulse into the cell body, and then axons carry the impulse away. So that's the direction of travel. So describe the neurons so you'll remember it. We're also going to be looking at skin. When we looked at stratified squamous epithelium, that is skin, and it's going to be at the end, so we can go back to our chart then. Before we do that, I just want to put epithelium and connective tissues together to show you how they work together. If I have epithelia sitting on a connective tissue, I usually call that a membrane, and we know from the first lab that there are 4 kinds of membranes. Serous membranes that line closed cavities, mucous membranes that line open cavities, cutaneous membrane, which is your skin or a dry membrane, and then finally synovial membranes, which I don't have on this picture, that line joint cavities. Synovial membranes are the only membrane that lacks the epithelium, so technically you can say it's not a true membrane, but they do put it in that category. So here's my picture of skin, and you'll notice that the microscope picture is stratified squamous. It's squamous at the top, many layers. So where the V is pointing, that's the epidermis, and where those 2 lines are at the bottom of that picture, that's where the dermis is. Now the artist drawing, you can see the epidermis is relatively thin compared to the dermis underneath that's thick. The dermis is where we find blood vessels, pressure receptors, nerve receptors, the oil glands, the sebaceous glands or sweat glands. We see the hair, which is really part of the epidermis, invaginates down into the dermis as well. Underneath the dermis, we have a fat layer that's called the hypodermis, but technically it is not part of skin. Skin has 2 layers, epidermis and dermis. So if I were to show you this picture under the microscope again and have an A at the top and a B at the bottom and ask you to label the dermis and epidermis, I hope you can. So what I want you to do now is I want you to go back through and look at all of my picture examples again, turn your paper over on the back and make an artist drawing from the micrograph or the microscope slide that's on each one of my slides. Fill that in. They should be the same color. Notice that they're usually either pink or purple or dark blue. Those are the 3 colors you're going to use, except for bone, which is kind of brown in color. So I want you to color them as you would see under the microscope, and on the lab practical I will have these same pictures, and you'll have to tell me what kind of tissue it is. After you have completed the tissue worksheet, then you'll submit it as a grade, and then don't forget to do your mastering. Now, next week, after we finish the tissues, you'll be taking your lab practical. There will be a review on Canvas that you can follow. Make sure you complete all of your MyLabs Plus before you take the practical. Review back through all your pre-labs and your worksheets as well. And this concludes the Tissue Lab.